Good morning, everybody. Welcome to the Tech Talk organized by VTT. Today we have a special guest from the University of Helsinki. I think this is the first time we have somebody from University of Helsinki, so I want more of this. Päivi Saavalainen is going to talk about microfluidics applications in biomedicine. I can't wait to hear about that. Päivi, floor is yours.
Thank you very much for the kind invitation. I'm really thrilled to be here talking about those, our more or less everyday equipments and techniques related to microfluidics, which we frequently use in biomedical field. My group is one of the 50 groups in our unit, research programs unit, which is one of the 3 units in Faculty of Medicine in Meilahti campus, and in our program in immunobiology we are about 8 to 10 groups. I'll go these techniques more or less quickly, but later on, I have some microfluidics chips and nanopore sequencers and stuff here too, so we can discuss them further. But the gel electrophoresis applications we do nowadays quite a lot with capillary systems, something about digital PCR devices based on droplets, nanowells, or other chip formats, microarrays, which are still used in some labs, which are now more or less replaced by next generation sequencing, and I'll go through those devices and platforms which are commonly used. And then my lab is especially focused on single cell RNA-seq, and I'll talk about those platforms. And also in protein levels there are new very interesting devices to do single cell proteomics. But first about this just everyday gel electrophoresis. Like in standard way we do it in agarose or polyacrylamide gels, separating the DNA fragments by size in the electrical field, and this is the typical gel electrophoresis picture. This is an old method, of course, and then the different sizes separate nicely in these gels. But it takes quite a lot of handwork making these gels, running them, and taking pictures. So for high throughput manner we usually use, and if we want to quantify very accurately and see the size very accurately of different bands or smears, we often use, there are several bioanalyzers by Agilent, where you can run 11 samples in the chip, and in this tiny capillaries structure in the chip, the gel electrophoresis takes place, and then you can see and quantify your PCR product, for example, very easily. In our lab we have Perkin Elmer solution, LabChip, which is capillary. There is a capillary which is picking up from the PCR products or libraries from the 96 well or 384 well plates directly one by one, so it's more high throughput than bioanalyzer. And here's the capillary which is sucking it from the chip, and then it runs, and the separation takes place here in that window. And also in our campus there is Agilent tape station, which is there is these tapes, screen tapes, which have the gels for various. You can use a couple of wells from here or the whole strip, and you can use the rest of the spots later within the next few weeks. So it's quite flexible for smaller number of samples. So the gel is already here in this tape, and the separation takes place there. Well, then people sometimes need to do digital PCR to get very, very sensitive, tiny amounts of the target, and if you need to quantify how many molecules of how many genomes of certain pathogen, for example, is there in the biological sample. You have to, the normal PCR for some rare templates is is not accurate enough or not sensitive enough. So you can divide the PCR volume and your template in hundreds of thousands or even millions of tiny droplets, and then do the PCR within the droplets. And Bio-Rad has this droplet digital PCR, has had that in the market for years already. And we have one or two machines at least in Meilahti for this. You can also do that same trick in nanowell capillary plates. Thermo Fisher has this format for digital PCR, so that there are 20,000 tiny wells in that chip. I have these chips here. We don't have the machine, I think nobody has in Finland, but we have been, for a material, we are interested in this nanocapillary plate, so we have bought only those chips to study their characteristics. So there are 60 micrometer hole-through wells in that chip, so that the PCR reaction takes place in each of these 20 wells, and then it measures the signal from every well. And another system is Fluidigm Biomark, where there is different kinds of chips for different applications. One of them is digital PCR as well, but also there is a quantitative PCR chips for 48 times 48 or 96 times 96. So you can put, let's say, 96 different patient or control RNA samples here, or cDNA samples, and then your 96 target genes, what you want to measure, measure from each of the 96 patients. So then it makes these 96 times 96 in almost 10,000 individual reaction chambers in this cassette, and measures the cycle by cycle the amplification of the target genes in each sample, and makes the quantification of those genes in each sample. And you can also use it for SNP genotyping and also single cell qPCR. So you have a cDNA library from 96 different cells, lysed and prepared one cell at the time in each 96 wells, and you put those cDNA libraries there, and then you measure 96 genes from every cell. Microarrays has been used for a long time, and there are still these scanners in our campus, but I'm not sure actually how much people still use it, because nowadays the Illumina sequencing is getting so cheap that the prices are quite compatible with microarrays. But the principle in microarray is that you have a synthesized or otherwise linked gene-specific probes, and tens of thousands or hundreds of thousands of them, even millions of them, in these chips. In each spot there is certain oligos only, and then you hybridize your DNA to that chip, and then the more there is certain gene, for example, presented or expressed in the sample, the higher is the signal, because these are using fluorescent labeled. The target DNA is fluorescently labeled first, so then you can measure the signals from the spots, and you can do it also with two colors from the same sample or sample mix. But then, well, sequencing has been, Sanger sequencing has been in place for decades, but now it has been replaced quite a lot with next generation sequencing methods. Most familiar to many of you is, of course, Illumina, which is the market leader still at the moment. So there you can do hybridize those. You put your PCR product or whole human genome, for example, in small pieces. You add their adapters, put them to the flow cell, where there are two types of adapter probes here, and then your target fragments bind there. You make a bridge amplification to make clusters of copies of each target, so that there are clusters formed and amplified. And then you do the sequencing for the whole flow cells. And for example, we have NextSec 500 in our unit. That gives from one flow cell run, it gives 400 million reads. So that means that there is at least 400 million of these spots in one flow cell. And then after you sequence, and there are, for every base, there are different fluorochrome labeled, and it measures cycle by cycle the synthesis in the sequencing reaction. And then from these images of the flow cells, after every nucleotide, it measures the signal, and that's how it reads the sequence of every single cluster, millions of those clusters in the cell. That's the basis of Illumina sequencing. And then these short sequence reads, you compare it to the reference genome. Let's say human genome sequence is here, and then computationally you align them, and you can then see their variants or count them. If you do it from the cDNA library, you can measure gene expression levels between patients and controls and so on. So Illumina is quite standard at the moment, and we have in FIM, there is HiSeq and MiSeq models, and in our unit we have MiSeq and NextSeq sequencers, and Viikkicampus has also Illumina machines. Viikkicampus has also PacBio, which is giving much longer reads than Illumina. Illumina reads has to be quite short. Maximum you can get 300 base pairs from both sides, so 300 plus 300. And the maximum length of this whole fragment in the flow cell is 1000 base pairs, because otherwise, if it's longer, then you don't get clear clusters, because the bridge amplification makes then otherwise too wide spots. So there is a size limitation in Illumina, but the PacBio is able to sequence huge, very long fragments, which is, for example, for de novo sequencing, if you want to analyze an unknown species where there is no reference genome even. It's easier if you can get the totally novel organism sequenced in longer pieces to make the map then. And that is based on that there is every molecule in the to be sequenced get to these tiny nano wells, where the synthesis of and the sequencing takes place one molecule at a time in these nanowells. And then similar to the PacBio, there is now this new technique of Oxford Nanopore sequencing. We have a couple of groups in Helsinki playing around with it. We have had this machine 2 years now. You can look at the flow cell I have here as well. So this is a kind of we felt like first that it's just a toy and it's fun to play around, but I'm actually taking it more seriously now, because it's getting better all the time. It still makes lots of errors, but so does PacBio as well. And PacBio is a €500,000 machine, the size of half an office room or something. And this is practically, the starter kit was €1,000, and this is the size. It's a USB stick which you put to your computer and just start sequencing. It's real-time sequencing. So you make your library, you have to put there certain adapters, but in your target library. But once you put it there, there is a tiny membrane here, biological membrane with biological pore proteins which are so tiny, there's so tiny hole that only single-stranded DNA can go through. So you have a double-stranded DNA as a template with certain adapters, and then there's a helicase enzyme here which is opening up the two-stranded DNA, and it pushes through the single-stranded DNA. And while it's going through the channel, it reads the sequence which is based on the electric currents changes in the flow of electrodes here. Because depending on which of the 4 nucleotides is exactly now going through, it changes the currency and the stream. It's very promising now, and now they have different models. They have advertised that the next model is actually even smaller, which you can put to your mobile phone. So it's actually very cool, and it makes also long reads. It's not that high throughput yet, so typically we get 400 million reads from every Illumina run, and this gives only something like maybe 500,000 sequences per run, but they are longer. It still makes much more sequencing errors, but I mean for us, we don't even want to sequence the whole human genome. With this, we are only interested on sequence some very difficult genes. So if we only amplify certain genes only, and we can get the read cover very high, so that we can sequence it thousands of times, and then we can see from the data that where are the sequencing errors, and we can make first the consensus, correct the errors, and then align that consensus to the reference. So it's not that big problem with this error rate. And in beginning, it was almost 30% of the nucleotides were wrong, so every third base was wrong. Now it's about 10%, and it's getting better all the time. But then to the single cell applications, which my group is developing. Instead of bulk RNA-seq, where we take let's say a piece of skin or a biopsy from the gut or blood sample with millions of cells, then we lyse them as a bulk, and we measure the RNA expression and try to find out differences between patients and controls, for example. But the tissues are very heterogeneous. It's a mixture of different types of cells, and when we do it in the bulk, we only measure the average of all these cell types, and which is not very accurate. So now there is a huge interest on doing this in single cell level, so that from every single cell we measure all the genes expressed there, for example, and we can do it for thousands of single cells separately. And the resolution this demonstrates that when you do it in single cell level, in bulks of batches of 10 cells each or 100 cells each, so even with 100 cell batches, you can't see any difference anymore between the batches, but in the single cell profiles you do see. So bulk RNA-seq and single cell RNA-seq are otherwise the similar protocols to make the study, except that in the beginning, so instead of lysing the whole tissue piece or million cells from the blood, you first have to capture them single cell by cell in 96 well plates. You can do manual picking or fax sorting to every one cell in a well, or then use this automatic cell capture devices. I'll go to this in detail, and then lyse and somehow tag them so that then you later know that which cell, where did each Illumina read from which cells they came. But after that, I mean, everything is pretty much the same. So you have to make the reverse transcription from the RNA molecule to complementary cDNA molecule. You can use their molecular identifiers to get rid of the PCR bias later on. You can put their spike in synthetic RNAs to help in analysis. And then you usually have to amplify it, the cDNA library, because from every single cell they have so tiny amount of RNA, so you have to amplify the full-length cDNA library. And for Illumina you have to shorten it, because as I said, the Illumina has the size limit of 1000 base pairs about. So you have to fragment and add the Illumina adapters to the ends of each fragment, or you can do a tagmentation with its transposase enzyme, which is cutting and ligating the adapters at the same time. And then it's ready for sequencing after some purification steps, and then you sequence it and align it to your transcriptome or genome maps of human, for example, or mouse, and then you read those counts and normalize and run different. Then you can do the heat maps and try to find different cell types from the data. This Fluidigm C1 was the first one in the market. Now I think VTT group has this machine, and also in Anna Vähärautio's lab in Biomedicine we have one. So it has been in market for a couple of years now, a bit more I think. So the first chip was the 96 cell chip, so that the cell suspensions go through these quite complex channels, and once there's one cell captured here, the rest of the cells pass by and go through to the next capturing site, and so until all of the or most of the capture sites are filled, then it's ready for then the rest of the cells are washed away, and then they move to separate chambers for lysis and cDNA synthesis in the reverse transcription, and you can add here the different reaction mixtures to those tiny chambers for each cell, and then in the end you take out there's output wells for the 96 different amplified cDNA libraries, and then you have to do the indexing of them and adding the barcode afterwards in just outside of the chip in the 96 well plate. The instrument is I don't know the current price, but some time ago it was 170,000 euro, and now they have also the high throughput chip for 800 cells, so that they can do 20 times 40 pooling here, and it's about a few euros per cell doing those analyses. The next one coming to the market after Fluidigm was WaferGen. They have this kind of nanowell chips of 72 times 72 wells. There is a bit more than 5,000, almost 5,200 tiny wells. And they have a nano dispenser, which is kind of a pipetting robot, which is putting those cell dilution there and the reagents to each of these wells. And then there is an imaging station which is scanning the chip and recording that whether there is one or more cell in each of the wells, and whether it's living or dead cell. So then based on that, this program makes a map to the dispenser, and you put the chip back there, and then only for those wells where you have one living cell, and one only, not 0, not 2, 3, just one. Then it pipettes the next step reagents only to those wells, and therefore, I mean, the data is quite clean, because there is no duplet wells or dead cells analyzed at all. But then 2 years ago there was a paper in Cell, or actually 2 of them, making nanoliter droplets and droplet-based capture of the single cell, which is really high throughput and less expensive as well. So the principle is, and my lab has now installed this system with dolomite pumps to our lab. So the principle is that we have 3 pumps here, which are pumping to this tiny chip 3 liquids. So barcoded beads, which come in a lysis buffer. One pump pumps that. Another pump pumps cells, just in PBS, BSA. And then the third pump pumps oil to the chip. And now these cells and beads are randomly entering to these droplets. So many of the droplets are just empty. In some of them there is only bead. Some of them have only cell. But a small fraction of the droplets have 1 bead and 1 cell. And because the beads come with a lysis buffer, the cell is lysed right away in the droplet. And then this bead is capturing the poly-A-RNA. And that's all what happens in the chip. And later on, we just break the emulsion and make the reverse transcription in the bulk. But because these beads have a unique barcode, then all the RNA is labeled with that bead barcode, and that's the way to track them later on from the data, that which cell had which genes expressed. Yeah, this is just we have a high-speed microscope with which we can monitor the chip. So 3 tubings are going into the chip, then the droplets are formed, and then the 4th channel is collecting the emulsion just to the Falcon tube. That's the chip, and we can accurately change the flow rates and pressures in all of those 3 pumps and monitor these chips all the time. There's a nice video in the internet how it looks when it slows down. So here is the bead coming. It's now there, and then there is a cell coming there, and then entering to here. But it's very rapid. Our system makes 4,000 droplets per second, so you can hardly see any. You can quickly see the beads are going there, and little hint of, okay, there was maybe cell, but it's very rapid. So we run through, 180,000 cells and 180,000 beads we put there, and it takes 15 minutes to run them through. And the emulsion looks like this. So normally we don't see cells anymore because they are lysed right away, but here in this experiment we didn't use the lysis buffer, so you can still see the cells and the beads. So the beads have a unique sequence. Hundreds of millions of these oligos in each bead. So there is a 12 base pair barcode, and then also another random barcode to eliminate the PCR bias from the data. But this is the bead specific barcode of 12 base pair. And if you think 4 to the power of 12 is about 17 million different sequences can be created with this barcoding. So every bead has millions of copies of the same oligo with the same barcode. And then after the barcode there is the poly T, and once this bead ends up to a droplet with 1 cell, which is then lysed, then the poly-A-RNA is captured with this T end, and that's all what happens within the droplets. Then we wash the oil away, break it, and wash the beads, and then we do everything, the rest in the Eppendorf tube. We do the reverse transcription, the first cDNA synthesis, then we amplify the cDNA, then we do tagmentation to cut them to illumina pieces and ligate the adapters, and purify in many steps, quantify it in many steps. We run this capillary electrophoresis to see the sizes, is it correct, and then it's ready for sequencing. So we first read only read 1 reads only these barcodes, 20 base pairs altogether, and then the read 2 comes from the other side, which comes from the real gene sequence. There is also other commercial droplet-based platforms now on market. 10x Genomics has launched the Chromium system. FIM in our campus bought this system, so the chip looks like this. I have one here as well. You can run 8 samples at parallel. So there is one chamber for oil, one for beads, one for cells, and then between these lines there is the microfluidic channels to make the droplets, and then you collect the emulsion here, and then you go further. So it's basically just more high throughput and automatized version of the dropsec. Illumina and Biorad together launched just very recently a similar one called DDSec, about 60,000 euro machine. The principle is exactly the same. And we have the system and pumps from Dolomite Bio. It's a UK company, and it's more handwork and more manual work than these push-a-button systems. But I mean, it's also more flexible, and they have hundreds of different chips for compatible with their adapters and pumps systems. So later on, we already ordered a droplet merging chip. I have it here as well. We have an idea to kind of, because here you can merge or inject actually more reagents to each droplet, so that then we could do later steps in PCR also in those droplets. We are testing that soon. And also that you can do double emulsion with certain chips, so that you can run these water in oil droplets again through water phase, so that then you have water in oil in water droplets, and those double emulsions you could run in the flow cytometer, for example. Or you can encapsulate cells in gel beads and culture them in these fluorocarbon oils, because it passes through the oxygen and so on. So it's more flexible, and there are more you can make. We have now together with Sami Francila's group here, we have done. My postdoc has been working here in the clean rooms and made our own PDMS chips for the system, because they are much cheaper. The Dolomite glass chip is 800 euros, and it's only €1 when we do it ourselves here. Then after RNA-seq in single cell, you have probably found all kinds of interesting novel cell types or genes, novel gene markers for different cells, and then you usually need to go to validate that in protein level, whether the expression is also true in protein level in each cell. So there is at least 3 protein level single cell devices available. One of them is, of course, the good old flow cytometer, which has been studied in immunology and blood research and whatever cell research for decades already. So in a flow cytometry, you can either analyze or even separate the facts, sort it with so you label the cells, intact cells, with antibodies with different fluorochromes, and if the cell is expressing in their surface, or you can stain also intracellularly your protein of interest, then the antibody is binding that, and when it's shining when it goes through the passes by the laser, then you can detect how big is the expression of the certain protein in each of these cells. You can analyze millions of cells with this, and you can combine. We have this Becton Dickinson Fortessa in our department. You can simultaneously measure about 14 different antibodies with that. We have 4 laser system there, and that's very pretty standard way to do single cell protein analysis. But then the more new ones are mass cytometry. Viikki campus has, I don't know if they have already got this, but at least they have had funding to buy this. So it takes place pretty soon, if not yet there. So this Helios Cytoph is Fluidim's machine. So here's the principle is otherwise the same, but not so that you put the cells and you have antibodies against your protein targets, but instead of fluorochromes, there are some stable metal isotopes conjugated to these antibodies. And therefore you can multiplex much more different labels to the same staining, because these metal isotopes you don't have such a compensation problems like you have in FACS, because you don't need to care about the wavelength overlaps of these different fluorochromes that you need in FACS, but because these metal isotopes are very accurate in size. So you label them, the antibodies, with these metal isotopes, easily 40, 50, 60 different ones. Those panels can be built. And then you shut them through, and when the cell is breaking and then this time of flight mass spec takes place, then these isotopes are separated by their size and read there and quantified from each cell. Then a very recent one, which we actually yesterday in our department, we decided to buy this, or at least test it first, if other campuses will put also some money on that. It's 55,000 euro scanner. It's single cell Western blotting, which is brand new technique. So you can capture there about 1,000 cells in these chips. There are tiny holes fitting one cell each. Then you wash the rest away. And then there's an SDS page, just a standard protein separation in electrophoresis, which takes place from each lysed cell here. And so in that way you can separate the protein of interest by size, because that's something that you can't see in the FACS or in the cell cytometer. So you do the Western blot, and then you blot it, and then you stain up to 4 different antibodies for your genes of interest. So for example, here there are 3 cells, and this cell obviously has this expresses only the exon 1 isoform of the protein. This cell is expressing the exon 2 isoform, a shorter protein, and this 3rd cell is expressing both of them. And then you can count them very accurately. And in the conventional Western, when you do it as a bulk, you have a sample of millions of cells, and then you do the Western blot gel. You can only see that there are two isoforms, but you have no idea that did all of the cells express both of them, or some of the cells these and some of the cells here. But you can really now, with this Milo by ProteinSimple, you can do in single cell level these isoforms as well. So this is something that we will definitely test, and if it's good enough, we will buy it. Then in the end, I will also show we have together with Joksa here and Sami, and then Lappeenranta Technical University, we have a new TECHES project from Challenge Finland competition running. The funding runs this year and next year. So we have a consortium for micropumps for single cell analysis and medical research. So Savonlinna group from Lappeenranta Technical University, Kari Ullakko's group, has this very clever magnetic shape memory metal micropumps, where you can have their tiny layer of in those microfluidic chambers or in these channels. You can place tiny pieces of that metal, and when you have that chip on the top of a magnet, which is rolling. Then this rolling magnet makes this metal layer as the peristaltic pump, and very accurate, very fast. So the Kari Ulleko's team is building us up here, and we want to replace our expensive dolomite pump systems with these chip integrated micro pumps, which can be so cheap that we can have them as disposable ones integrated into the chips. So the prototypes of the chips we are doing with Joksa and Sami here, and then we have also companies involved. So Perkin Elmer is of course a big global company having lots of research activities in Turku site. And then we have 2 bioinformatics companies involved making user-friendly data analysis tools for single-cell data. We have UPM, who are interested on the Grodex hydrogel applications in microfluidics. And then we have also machine vision and mobile microscopy techniques by Fimmig and Grundium, so that we could kind of on the chip analyze what's happening. And we apply this also to diagnostic applications. And Ginolis is an Oulu-based company in this chip side. So very interesting projects going on microfluidics, and I'm very happy to have launch quite intensive and very fruitful collaborations with Aalto. Very fun to learn totally new things, and seeing that actually only a few kilometers away from Meilahti, I mean, we can do our own chips here and bring our own ideas that what we need for biomedicine. People are here in Otaniemi to help us, and this has opened my eyes to these powerful collaborations, and we should really bring these connections and collaboration networks up further in Helsinki area, I think. Thank you very much for your attention.
Wow, an impressive technology review, I must say. You can even fabricate your chips if you make a spin-off here as well.
Yeah.
I'm sure we have questions back there.
Okay, so you have many methods and many devices, but is this somehow clear that how do you know how many errors you are making in the analysis, and is this error correction somehow mathematically demanding?
For the sequence errors,
Yes.
or? Yeah, so when we cover, for example, one gene, I mean thousands of reads coming from the same gene, and we see the sequence, we can easily see that what is real polymorphism, because if it's polymorphism in our germline DNA, then half of the reads should have the other allele and half of the other one. But if there's just random mismatches somewhere there, then it must be sequencing error. So if we cover it, for example, with Nanopore, we have to sequence it very deeply, the same sequence again and again and again, and then we can see from the data that there is some sporadic kind of errors every there and there, so that they must come from the sequencer, and we can correct that to the consensus. Yes.
Other questions? You seem to have a lot of equipment, or I don't have the feeling that you are lacking equipment, but maybe I'm wrong. With all that equipment, do you generate some new processes or some intellectual property, some new devices that are some of you guys in the lab want to take into commercialization?
Well, most of those, I mean, those capillary electrophoresis stuff, we just buy the kits and we just run them and we use them as such. We don't really innovate anything new to those commercial platforms. But for example, in the single cell field, we have lots of ideas to how to improve that, making it cheaper, making it better, and how to build better chips to capture certain cells only, for example. And so I think I hope that from this TECHES consortium we can have a new IPR created, and who knows, spin-offs as well. But I mean, but at least we have good companies collaborating us, and maybe they can build up also new products with our help.
So would you say this is the Finnish signature in this field is single cell handling? Is that?
Well, Finnish signature?
I mean, yes, the competitive edge of Finland in this field, or of your lab, let's say.
Well, maybe my lab was the first, among the first ones trying these technologies some years back. And I also went to Karolinska for sabbatical to learn, because at that time, 3-4 years ago, we didn't have any single machine yet. So I went to learn those techniques here, because Karolinska is one of the leaders worldwide. They have been, the pioneers have been there, and there are very much activities in US and Israel, for example. So there are new devices coming. So no, we are not the first ones, not at all. But I mean, we have our own ideas to how to improve certain applications, and especially my interest is in immunology.
Right.
Yeah.
So we can look forward and see maybe some
we'll
new
see.
companies in
We'll
the future
see.
in Finland.
Yeah.
Other questions? No question? I have a last one. We see that you miniaturize the devices are miniaturized more and more, and we can understand why. But is there a need to go further? And if yes, why?
Well, I think now when we think of single cell stuff, I mean the cells will always be the same size. So I think for those cell diameters, I mean I think this is now probably the smallest we can go. Of course, we can make it more high throughput and have and capture efficiency can be improved. For example, now in the droplet-based methods, only a few % of the cells enter to the droplet with the bead, and that's where we get the library done. So we can do it for a few thousand cells from each run. But I mean, I think the sequencing costs are also quite expensive still. So usually people don't afford to sequence more than a few thousand cells per experiment, because it otherwise too expensive, the Illumina sequencing. But I can see that when hopefully the Illumina lowers the price at some point, so that the sequencing price is not the bottleneck anymore, then I think there's good place for these very high throughput devices with capturing of hundreds of thousands cells and making libraries of them. But it's all about money, I think, at the moment.
Maybe in the future, during the first date, a man and a woman will sequence each other to see if they are compatible. You have a question here.
Yeah, Erkki Seppälä from Skunk Developments. Okay, as we see, there are commercial equipment already available, and now from engineering point of view, the question is that okay, they have competitive edge because they are already with a ready product in the market. How do you see our possibility in Finland to develop something that would be lower cost, would be better in the market? Because I mean, as you said, Illumina will bring cheaper equipment. If we start developing now, where are we 2 years from now when Illumina has already the next cheaper version? How do you feel the potential of developing own devices here in Finland?
Well, I think, for example, the droplet technology, now all the Illumina Biorad, the 10X, they had the exactly same principle, which was copied from the academic Harvard paper, the DropSec, which we adapted as a kind of cheaper platform, but more laborious. But basically now, I think now there is a competition and this, because these similar devices are coming from other companies as well. So now the price level in the droplet machines is about that 50,000-60,000. But basically they are just boxes of simple pump pushing the pressure. So I think for such a box, I think it's quite high price. So I mean, with the, for example, micro pumps, I mean, we can do, if we can do it and the chip costing like just €1, I think there is something to think about. What is the real cost of these devices? Of course, you have to think about optimizing and making the reagent kits and stuff. Though it's not, you can't sell it with €1. But I mean, chips are not expensive, and pumps are just pumps.
Yeah, because okay, it would be of course nice, as Philip said, to bring some technology companies in Finland.
Yeah.
Okay.
I hope to be involved in that.
Well, we certainly have the right ecosystem.
Yes.
I think so. A last question? Yes, here.
Okay, the devices start to look like a consumer products, but what could a normal consumer do with these devices? I mean, for example, the mobile version of this. So, is there a useful application for?
Not perhaps for any customers who have it. I think the customers for those mobile phone sequencers are also researchers who probably need to go to a jungle and sequence on site Ebola or something. And you can't bring the Illumina machine to jungle, and you have to do the sample prep in somewhere in the very rural areas if you want to do some research on site. So I think that mobility action, but then it's of course a different thing that do you have a mobile phone kind of how do you connect in the jungle? And then actually the data files are huge. So now already with this, I mean, we have been struggling with the size of the files. So if you go to sequence something in jungle, you have to have a backpack full of in external hard drives because you need so much computing space. So.
And another one for the battery, I guess.
Yeah.
Other question? Last question? Well, if not, thank you again for your nice presentation. Thank you, Päivi.
Thank you. And if you want to see these, I mean, just come.